Welcome back, friends. In my previous video, we discussed about the structure of polysaccharides, and now let's proceed with cellulose. Cellulose is the polymer of beta glucose, so it's different from starch and glycogen in the fact that starch and glycogen, they are polymers of alpha glucose, while cellulose is the polymer of beta glucose. So unlike starch and glycogen, it has a structural rule. So starch and glycogen, they have storage rule, but cellulose has structural rule. Now, when two molecules of beta glucose line up, the hydroxyl group on carbon atom 1 can only line up alongside the hydroxyl group on carbon atom 4 if the molecule is rotated at 180 to the other. This is because the hydroxyl group on carbon atom 1 projects below the ring, and the hydroxyl group at carbon atom 4 projects above the ring. So now assume maybe you have two beta glucose molecule. Maybe this is your first beta glucose molecule, and here is your first carbon, second, third, fourth, fifth, and then the sixth carbon molecule there. And then you have another beta glucose molecule, the first, the second, the third, the fourth, the fifth, and then the sixth carbon. So what we are saying here is that if here, according to this information, according to this statement, that this is because the hydroxyl group at carbon atom 1 projects below the ring. So at carbon atom 1, the hydroxyl group projects below the ring. Hydroxyl group, then hydrogen. Where at carbon atom 4, it projects above the ring. Hydroxyl, then hydrogen. So the same condition takes place here on hydroxyl, hydrogen. So let's call this the first and this is the second beta carbon atoms, and then we want the hydroxyl group at carbon atom 1 of this first molecule to project on the same side as the hydroxyl group of this carbon atom 4. So what will happen here is that this molecule should rotate. It should rotate this way 180 degrees, so as this hydroxyl group projects below. So the resulting molecule will appear like this: the first, the second, the third, the fourth, and here is hydroxyl, hydrogen, then hydroxyl, hydrogen. So after rotation of this molecule, after rotation of this second molecule, then the molecule will appear like this. Oxygen will be here, hydroxyl, hydrogen, and then here will be our sixth, our first, our second, our third, our fourth, our fifth, and our sixth carbon. And here it will be hydrogen, then hydroxyl. So the coming molecule, this is the first, the second. The coming molecule, we want these to project on one side, and as you can see here, the hydroxyl group also of this molecule projects on the same side as the hydroxyl group. So the hydroxyl group, according to this sentence, when two molecules of beta glucose line up, the hydroxyl group on carbon atom 1 can only line up along the side of hydroxyl group on carbon atom 4 if one of the molecule is rotated. So you rotate, unairotate one molecule 180 to the other. Kona molecule inayofuata hapo kwa cellulose inabidi ifanye hivyo. Hiyo hapo tunafanya ili hydroxyl group zikae upande mmoja na ziweze kufanya hydrogen bonding, ambayo hapo itapelekea cross linkage. So this rotation of successive residue is the underlying reason why cellulose has a different structure to starch. So cellulose has a different structure to starch because of the rotation of rotation of the molecules. And as you can see here, it is the most abundant organic molecule on Earth. Cellulose is the most abundant organic molecule on Earth. It is constituent of all plant cell wall. Constituent of all plant cell wall. It is abundant in plants, although it is found in some non-vertebrate animal and a central fungi. It is abundant is due to its being a structural component of all plant cell, making up about 20 to 40% of the wall on average. The structure of molecule reveals its suitability for this rule. It consists of long chain of glucose residue with about 10,000 residue per chain. The beta 1,4 linkages make the chain straight. The beta 1,4 linkages make the chain straight, in contrast to starch, where the alpha 1,4 linkage cause the chain to be curved or helical. So in alpha 1,4 linkages, they cause the chain to be curved, while the beta 1,4 linkage, they cause the chain to be straight. Hydroxyl groups projects outward from each chain in all directions and form hydrogen bonds with a neighboring chain. So this cross-linking bind the chain rigidly together. The absence of side chain allows these linear molecules to lie close together. So presence of hydrogen bond allows the cross-linkage, and absence of side chain allows the linear molecule to lie close together, as you shall discuss in your Form 6 polymerization and the structures of different types of polymers. So structure of cellulose, it consists of straight chains of beta glucose molecules, where the hydroxyl groups projects on both sides of the chain at alternating positions. So the free hydroxyl groups are thus in exposed positions for hydrogen bonding with neighboring hydroxyl groups of other chain, which result in the formation of bundles of cross-linked parallel chains as illustrated below. This structural property combines strength of the glycosidic bond and hydrogen bonds to make cellulose such a strong polysaccharide suitable for conferring strength in cell wall of plant. So cellulose has two bonds, which are glycosidic and hydrogen bond. So the structure of cellulose can be seen as in this diagram here. So also in the plant cell wall, we have different materials such as suberin, lignin, calcium pectate, magnesium pectate, and hemicellulose, together with hydrogen bonds combined to provide strength cell wall. So in cellulose, we have straight chains made up by 1,4 beta 1,4 glycosidic bond, and then we have hydrogen bonds at alternating positions, making the close linkage in the structure of cellulose. So this is how cellulose occur, and as you can see here, this is the shape or structure of cellulose with close linkage, while this is the shape of starch molecule. It is just a spiral. So function of cellulose, kwamba cellulose ina kazi gani? Kwanza kwenye plant cell wall ina prevent bursting ya cell isipasuke kama maji mengi yataingia by osmosis. Lakini ya pili, turbidity ya cell, kwamba baadhi ya mimea yenyewe haina miti, kwa mfano ya nyasi ndogo kabisa, tunazita sebaceous plants. Sebaceous plants, they only use turbidity of the cell to support them. Lakini pia it allow proper function of the plant cell. Because cellulose cell wall is permeable to different materials to enter and leave the plant cell. So that's why we are saying that it allows the efficient functioning of the plant cell. Also, it is an important food source for some animals, bacteria and fungi. Human beings, they are unable to digest cellulose but some of the animals, bacteria and fungi, they have cellulose enzyme which can digest the cellulose. So for them, it's the source of food. And this question is normally asked by the teachers, the commercial uses of cellulose. So first, it is the raw material in manufacture of many industrial products, such as paper, iron, and plastics, raw material. Second, cellulose derivatives such as cellulose nitrate are used in manufacture of films and explosives. The third, cotton, which is the pure form of cellulose. Kwa ile pamba ambayo unaiona ni cellulose kabisa, cellulose pure 100% is used in the manufacture of clothes. And the iron made from cellulose is used in the manufacture of industrial belt and the tire cords. So these, they are industrial uses of cellulose. Now from there, we have compounds closely related to polysaccharide. Compounds closely related to polysaccharides. So they are not directly polysaccharides, but they are closely related to polysaccharides. The first compound is chitin. Chitin is closely related to cellulose in structure and function, being a structural polysaccharide. So chitin it occurs in some fungi where it is fibrous in nature, contribute to the cell wall structure, and in some animal groups, particularly the arthropods, where it forms a special part of the exoskeleton. So structurally, it is identical to cellulose. Chitin is identical to cellulose, except that the hydroxyl group at carbon atom 2 is replaced by that group. So this group, we may call it as the amine, carboxyl, and then methyl. So I don't have the exact name of this group, but no need of this group at this time. But what you should know is that the hydroxyl group at carbon atom 2 is replaced by that group. And as you can see here, this group is here and this group is here, indicating the replacement of the hydroxyl group at carbon atom 2. So as we have seen that its function is to act as the structural carbohydrate in fungi and in some of the insects' exoskeleton. Let's jump to murine. Murine is a polysaccharide which acts as the strengthening material of bacterial cell wall. So murine is found in bacterial cell wall. It is similar in structure to chitin, containing nitrogen like chitin. So they differ little. And then we have inulin. Inulin, it occurs in roots and tubers of dandelion and small amount in dicotyledons. So it can be extracted as white powder soluble in warm water. It is hydrolyzed by dilute acids such as HCl or by inulase enzyme to form fructose only. So inulin is made up of only fructose. That's why on hydrolysis it releases only fructose. As you can see here, inulin, then inulase provides the fructose. Another compound is hemicellulose. Hemicellulose differs from cellulose in that they are easily hydrolyzed by boiling with dilute acids. The products are the mixture of exosomes and pentoses. So as we know that cellulose, they are made up of only glucose, beta glucose, which are exosomes, while the hemicellulose is made up of both pentoses and exosomes. So it forms a part of cell wall and may be food storage substance such as in coffee and beans. Kwa kwenye kahawa na marage, hemicellulose is food storage substance. During germination, they yield sugar by the hydrolysis of enzyme hemicellulose. So that's the end of the compounds related to polysaccharides. And now let's jump to the functions of polysaccharides in general. The first function is the structural functions. Some polysaccharides are the components of the cartilage, bone, cornea, and so all of those polysaccharides, they act as the structural function. Also, some are antigen in gram-positive bacteria. So what are the antigen? Antigen, they are structures in the cell wall of bacteria which can cause or can stimulate the production of antibodies. So you'll understand well later on in your university level what are the antibodies when you will be studying about immunity. And some they are components of the cell wall of prokaryotic and plant cells such as murine and cellulose. Some form cell wall matrix in the plant. Here we are talking of cellulose. And then the second major function, protective function. Some polysaccharides are anticoagulant in mammalian blood tissue. Anticoagulants is in molecule ambazo zina prevent blood coagulation. Blood coagulation ni kuganda kwa damu. Kwa kama hizo polysaccharides zisingekuwepo, damu ingekuwa inaganda kwenye mishipa. Na ikiganda kwenye mishipa mtu aweza kufanya chochote kwa angekuwa anakufa. So that is the protective function. Pia some form gel which retain water for drought resistance such as mucilage. Kwa mucilage kwa baadhi ya protoctist yenyewe ina retain water in drought, and that is the protective function. Then we have food storage compounds which are in some plants they store food such as hemicellulose, starch, and even glycogen. So this question is in the biological science book. What structural features of carbohydrate account for the fact that wide variety of polysaccharide exists? So most of times students, they don't understand this question, that they read the answers in the biological science book, but they don't understand the fact behind. So the fact behind this question is that we have monosaccharides, and the monosaccharides, they have different features. The different features of monosaccharides and the ability of the monosaccharides to combine to form different bonds, to form different length of the chain, makes different variety of polysaccharides. So monosaccharides, they do different maneuvers to form disaccharides, and then disaccharides to join and to form polysaccharides. So as you can see the points here, first, both pentose and hexoses can be used to make polysaccharides, although normally only one type of monosaccharide is used, but sometimes can be used both, example in hemicellulose. The second thing, two types of linkage, 1,4 and 1,6, and 1,6 allow the branching to occur. The third thing, length of the chain in the branches and the extent of branching can vary enormously. For example, we know that amylose has a short length, while amylopectin has a long length, while the amylose has no branching, amylopectin has branching. While the extent of branching is that amylopectin has less branching as compared to glycogen. So you can see that difference. Then the fourth point, alpha and beta forms of monosaccharides, they are important. As you can see that starch is formed by the alpha glucose, and it is spiral, is used for storage, while the cellulose is formed by the beta glucose. It is straight chain and it's used for structural function. Then sugars may be ketoses or aldoses. So sugar being ketose or aldose also makes the structure of polysaccharides or type of polysaccharide to differ. The last point, the high chemical reactivity of the sugar, which are aldehyde, ketone, and hydroxyl group. So since these groups are more reactive, they can react with different molecules and to form different kinds of carbohydrates. So the last study question here is that what is the condensation reaction and what is the similarities between glycogen and starch? I think that's not the last. The last is that why human being cannot digest cellulose. So cellulose, why human being we don't digest cellulose? Because we lack the enzyme We lack the enzyme cellulose, which can hydrolyze cellulose to form glucose and fructose. Cellulose to glucose to beta glucose. This is made by the beta glucose. And about the similarities of glycogen and starch, as you can see here, both are polysaccharide made up of alpha glucose residue. Both have 1,4 glycosidic bonds together with branches of 1,6, and both are storage form in living tissue. So in the next lecture, we shall be discussing about lipids. Stay with me, stay tuned, subscribe the channel, share the videos to your friends, and tell them to subscribe the channel. Also, don't forget to click the link below to join my Telegram group for more discussions and questions. For any comment, just comment below, and then I will answer your concern. Thank you everybody, and have nice studies.